Hi, good afternoon, everyone. So today we will summarize week eight's content, which was mostly about digestive system, the different organs, the glands therein, how food passes right from ingestion. It will basically start with the different modes of nutrition and then eventually the complete kind of the different glands, that its secretions and stuff, and how food is digested and absorbed and ejected out. Okay, so I will start sharing the screen. Then finally, we will look at some of the questions. Okay, so to begin with, uh, when we talk about nutrition, what is nutrition? Nutrition is basically the process by which living organisms utilize food, like eat and utilize food to carry out their life processes. Okay. The mode of nutrition describes how an organism acquires its food and the way it processes the nutrients. And based on the source and the method of obtaining food, nutrition can be very broadly categorized into autotrophic mode of nutrition and heterotrophic mode of nutrition. Now, the different modes of nutrition reflect the different ways in which organisms have adapted to their environment and availability of food resources. So, autotrophs, auto means self, right? So autotrophs serve as the foundation of ecosystems, supporting heterotrophs, which include herbivores, carnivores, omnivores. So these are all examples of heterotrophic organisms. Herbivores, carnivores, omnivores, parasites, and decomposers. Understanding these nutritional strategies is crucial for studying food webs, energy flow, and ecological interactions taking on around in our environment, in the nature. So, if we talk about autotrophy, which means self-feeding organisms, which synthesize organic molecules from inorganic sources. Autotrophic organisms can be photoautotrophs or chemoautotrophs. Photoautotrophs utilize, like they do photosynthesis and make organic compounds using energy derived from the sun or solar energy. Then there are chemoautotrophs, which do chemosynthesis, which make organic compounds using energy derived from the oxidation of chemicals. Then there's heterotrophy. There are other, they use, they feed, uh, they don't synthesize their own food, but they actually obtain organic molecules from other organisms. Okay. So these can be consumers which ingest organic matter which is living or recently killed, like herbivores, carnivores, or scavengers. There can be detritivores which ingest non-living organic matter like earthworms, woodlice. And there are saprotrophs which feed on non-living organic matter by secreting digestive enzymes and then absorbing the products. For example, fungi and bacteria. Now we will go through it one by one. Autotrophic organisms are capable of synthesizing their own food from inorganic substances like CO2 and water, uh, and using an external energy source like light. Okay, so these organisms are primary producers in ecosystems, forming the base of the food chain. So one is photosynthesis, photoautotrophs. This process is usually carried out by green plants, algae, and certain bacteria, which convert light energy, usually from the sun, into chemical energy stored in glucose. It involves the use of chlorophyll, a green pigment in chloroplasts, to capture light energy, and this energy is eventually used to convert CO2 and water into glucose and oxygen. Examples are definitely the different kinds of plants around us. Then cyanobacteria, scientific name is Anabena. Then there are algae like Chlamydomonas, etc. Then there's chemosynthesis, process by which certain bacteria synthesize organic compounds using the energy derived from the chemical oxidation of inorganic molecules rather than using sunlight as the source of energy. This mode of nutrition is common in environments where sunlight is not available, like deep sea hydrothermal vents. So, organisms that perform chemosynthesis use substances like hydrogen sulfide, ammonia, or methane as energy sources. Examples are chemosynthetic bacteria like Nitrosomonas, which are also involved in the nitrogen cycle going on in the atmosphere. It oxidizes ammonia to nitrites. Then we have heterotrophic organisms which cannot really synthesize their own food. Instead, they rely on other organisms like plants, animals, or dead organic matter for their nutritional requirements. Heterotrophic nutrition can be further classified based on the source and the method of food intake. One is holozoic nutrition. It involves the ingestion of solid organic material followed by the digestion, like breaking down of complex food into simpler forms, then absorption, then assimilation and egestion, which is the removal of undigested matter. This mode of nutrition is characteristic of most animals, holozoic mode of nutrition. So the different stages involved in holozoic nutrition are ingestion, where the food is taken in, then digestion, where it is broken down into smaller molecules by enzymatic action. Then there's absorption, uptake of nutrients into the bloodstream or cells. Then assimilation, utilizing the absorbed nutrients for energy, growth, and repair. And then egestion, which is the elimination of undigested waste material. Examples are definitely humans, mammals, reptiles, birds, and fishes. So also in amoeba, it also undergoes holozoic mode of nutrition. So what happens is this is the amoeba, okay, the cell, and suppose it is sensing some kind of uh, like it sensed the presence of food nearby. So amoeba senses food. Pseudopodia or false feet are kind of these extensions which eventually extend out of the body, and I mean cell body, and then pseudopodia surrounds the food, like these false feet eventually surround the food, and it's engulfed inside in a food vacuole. Here this is the food vacuole. Then enzymes are released from the cytoplasm and secreted into the food vacuole. Okay, food is digested and soluble materials are absorbed and assimilated or used up for growth and repair, and undigested waste is expelled outside. Okay. Then we have saprotrophic mode of nutrition, where the organisms obtain nutrients by breaking down and absorbing organic matter from dead and decaying organisms, and they secrete digestive enzymes into their surroundings to decompose complex organic substances into simpler molecules, which are then absorbed. So, saprotrophic organisms play a vital role in the ecosystem by recycling nutrients, for example, fungi like mushrooms, rhizopus, and certain bacteria. So, these are the different kinds of mushrooms which show saprotrophic mode of nutrition. Okay. Then we have parasitic mode of nutrition, where parasites obtain nutrients by living on or inside a host organism, often causing harm to the host. Parasites can be external ectoparasites or internal endoparasites. They absorb nutrients directly from the host's body, often causing disease or damage to the host. Examples are ectoparasites like lice and ticks live on the surface of the host, whereas endoparasites like tapeworms and plasmodium, which causes malaria, these parasites live inside the host's body, hence these are called endoparasites. Then we have symbiotic nutrition as well, where two different organisms live in close association and mutually benefit from each other. And this relationship can take various forms, including mutualism, where both the organisms involved in this interaction are benefiting from each other. It can be commensalism, where one organism benefits from the other, however, the other organism involved in the interaction is unaffected. Or it can be parasitism, where one benefits at the cost of the other organism. How we looked at here, the same thing. So, actually, parasitism is actually a form of um, like can be considered under symbiotic relationship where one is benefiting at the cost of the other. Okay. And so symbiosis often involves the exchange of nutrients between two organisms. So in mutualism, if we talk about where both the organisms are benefiting from each other, we have lichens, which is a symbiotic relationship between fungi and algae, where the algae provide food through photosynthesis and the fungi offer protection and nutrients. Then we have commensalism, where the two organisms involved, uh, out of the two organisms involved, one is benefiting from the other, however, the other, it doesn't really affect them at all. And for example, there are barnacles on whales, where barnacles gain mobility and access to food, whereas the whale is unaffected. So this is what is the pictorial representation, where symbiosis is the long-term association between two organisms of different species. We also have mutualism, where both partners benefit. Another example is nitrogen-fixing bacteria in the roots of leguminous plants. Then we have parasitism, where protozoans can live inside humans, where the protozoans are benefiting from the humans. However, the eventually it causes diseases in the organism in humans. Then we have commensalism as well. Okay. But this is not always true. A bird and a tree, like tree is providing shelter and stuff, but also some in some cases, like definitely trees providing food and shelter to the birds. However, suppose there are organisms, insects, bees, and stuff, which also in some cases in some plant species, these actually help in pollinating the plants. I mean, pollinating the seeds. Okay, so that the plant species is eventually propagated. Okay, it leads to pollination and flowering of the plants. So it can be like mostly this is the scenario where common birds are benefited. However, it does so organisms can also be a pollinator for certain plant species. Okay. Then if we talk about lichen symbiosis, it is a relationship, symbiotic relationship between algae and hyphae of a fungus. These are actually fungal hyphae here. Okay. And algae provides food for hyphae through photosynthesis. Algae is actually synthesizing the food. This one here, the green one here, algae is synthesizing the food, and hyphae provides a suitable environment for the algae to survive. And sometimes these can get kind of released or detached, uh, which are then called soridia, which are actually algae embedded in the clustered hyphae, these parts. Okay. And we were talking about commensalism, where one species is benefited while the other isn't affected. So this is a whale flipper, and then you see these ones, these are the barnacles on the uh, like hitchhiking on the way. Okay, so the barnacle actually benefits by finding a habitat where nutrients are available. Presence of barnacle populations doesn't really hamper or enhance the survival of the animals carrying them. So these actually act as kind of a hitchhiker where they get to move uh, from one place to the other, as well as they can get access to food resources as well. So why do we really need food? For energy, muscle movement, maintaining the temperature, carrying out the different metabolic reactions. Okay, definitely for growth. It gives us the individual materials for cell growth and for removal of aging cells, senescent cells, and also to keep the uh, body cells and body healthy organs, to keep the organs functioning properly. So when we talk about the anatomy of digestive system, if we look at the complete like overall portions or regions and organs involved are the buccal cavity, which again harbors different kinds of organs. Then there's esophagus, the food or the food pipe. Then comes the stomach, then the small intestine and large intestine. Okay, and this is the overall overview of the digestive system, starting with buccal cavity. So this is actually the mouth, which consists of a vestibule and oral cavity. The slit-like space between cheeks and gums is known as the vestibule, and the cavity is surrounded by palate. These ones, the upper roof, palate, tongue, and teeth, is known as oral cavity or buccal cavity. So mouth is the first passage of food where large piece of food is fragmented to small pieces with the help of teeth and mixed with saliva to form a bolus. So tongue manipulates the food during chewing and mixing with saliva. The mixture of food with saliva, the bolus, is then moved inward through pharynx into the esophagus or the food pipe. The process is called swallowing or deglutition. Okay. So if we talk about the roof palate, I mean the hard palate and soft palate, it is the roof of the oral cavity consisting of two parts. The one that we have immediately behind the teeth is the hard palate, and eventually as we move to like go down near the like near the tonsil area, this is the part where it's softer and it's called the soft palate. So hard palate is the anterior part that contains the bone. Soft palate is the posterior portion, this part here, that consists of skeletal muscle and connective tissue, this one. Then we have this small extension, uvula. This is the posterior portion of the soft palate. The palate separates the oral cavity from the nasal cavity and prevents food from passing into the nasal cavity during chewing and swallowing. Then we eventually have the tonsils, which are located in the lateral posterior walls of the oral cavity, in the nasopharynx, and in the posterior surface of the tongue. Now, when we talk about the tongue, so there are taste buds present. This is the tongue here. There are taste buds present. Okay. Now, if we look at a taste bud here, it is covered using different types. It is covered with different types of cells. So, taste buds are microscopic sensory organs embedded within the papillae of the tongue, housing gustatory receptor cells. Um, these ones, the gustatory receptor cells that detect sweet, sour, salty, bitter, and umami tastes. They are constantly renewed every 10 to 14 days, these taste buds, ensuring that the tongue maintains its sensitivity to flavor changes throughout life. So, circumvallate papilla are there, these ones. Okay. The circumvallate papilla is a large dome-shaped structure located at the back of the tongue, surrounded by a trench and containing numerous taste buds. These papilla are particularly sensitive to bitter tastes, playing a key role in detecting potentially harmful substances in food. Then we have filiform papillae, this one. It is conical thread-like, covering most of the tongue's surface, providing texture and aiding in food manipulation. Unlike other papillae, it lacks taste buds but contributes to the tongue's rough surface, enhancing grip during chewing. Then we have foliate papillae, this one. It consists of folds or ridges on the lateral edges of the tongue, housing taste buds that are sensitive to multiple taste modalities. These structures are more prominent in younger individuals and may eventually disappear with age, affecting taste perception. And we have taste hairs also, which are like microvilli. Okay. So, the taste hairs, which are like microvilli extending from the gustatory cells. These are the gustatory cells, and these are the taste hairs over here. Um, Increasing the surface area for chemical detection. They project into the taste pore where they interact with dissolved food molecules to initiate the taste signals. This is the taste pore. Taste hairs are eventually projecting here and then interacting with the food molecules. Okay. Then there's transitional cell as well. It serves as an intermediate stage between basal cells and mature gustatory cells, supporting the renewal process within the taste bud. It gradually differentiates into specialized cells, contributing to the taste bud's dynamic structure. Then you have gustatory cells, which is a specialized receptor cells within the taste bud that detects chemical stimuli and transmits taste information to sensory neurons. It contains taste hairs that extend to the taste pore where they bind with taste molecules to trigger neural signals. Now, taste pore is a small opening, this one here, on the surface of a taste bud, allowing saliva and dissolved food particles to reach the taste hairs. This gateway is essential for the transduction of chemical stimuli into electrical signals within the gustatory cells. And basal cell is there, this one is the stem cell located at the base of the taste bud. This is the complete taste bud, okay? And at the base is the at the base are basal cells, which are stem cells, which can regenerate gustatory and transitional cells. Its continuous division ensures that the taste buds functional longevity and adaptability to environmental changes. Okay. Then if we talk about the anatomy of the tooth, uh, teeth are the hardest tissue in our bodies, and they are only for chewing, but also for supporting our cheeks and lips, and it also helps in determining the shape of the face, and it also helps in pronunciation. Okay. So tooth consists of this part, which is the crown, the upper visible part of the tooth. Then comes the enamel, this one here, this layer. This is the hard, shiny white substance covering the tooth. The enamel protects the dentin beneath the crown. Then comes the gingival sulcus. This part here, this part. This is the fold between the tooth and the gum where dental plaque and tartar may accumulate. Then comes the dentin, this part here. Okay, this layer here, which is the hard tissue covered with enamel, and it forms the body of the tooth and surrounds the dental pulp. Dentin is what causes the teeth to appear yellow, especially when the enamel layer is thin. Then we have the dental pulp, which is very sensitive. The nerve is there, the soft tissue in the middle of the tooth containing large amounts of nerve tissue and blood vessels. The pulp contains the nerve ends that transmit pain signals. Then eventually we have the root here, these ones. The portion of the tooth surrounded by the alveolar bone. The root is covered with cementum, a substance that gives it protection and serves as an anchor for the ligament that holds the tooth into the bone. And then there are different types of teeth serving different functions. The very first one is the incisor, which in case of humans, we see it directly in front, is the front part of the dental arch, front of the mouth, has just one root, and it is usually used to bite off food. Then we have canine, the sharper ones, right beside the incisors, between the lateral incisors and the first premolars. It also has just one root, and it's a little this one here should be a little sharper, used to tear and rip the food item apart. Then there's premolar. This is between the canine and the first molars. Generally has one root, but this has cusp and a little more flattened. This is used to grind the food items. And finally, we have molars. It is present at the rear part of the dental arch or present at the back of the mouth, has several roots, and this is also used to grind the food, and it has like it has many cusps and much larger surface area. Okay. So in general, incisors are used for cutting, canine is used for tearing the food, molars, premolar is for grinding, molars are for crushing the food item. So this is the formula for the like number and the placement of the different types of teeth inside the buccal cavities of different organisms. Okay. And humans have 2 1 2 3 formula. Okay, usually, and others can have variable amounts as you see here for rabbits, cows, and sheep. Now, then we move on to the food pipe or the esophagus. It is also known as a gullet. It is about 25 centimeters long and is present behind the trachea and the heart. Its primary function is to conduct the food from the pharynx to the stomach. Food in the food pipe is pushed downwards by involuntary muscle contraction of circular muscle, and this particular movement is called peristalsis. And due to the contraction of the longitudinal muscles, lower part of the food pipe becomes short, which eventually pushes its wall outward so that it can receive the bolus. The circular muscles of esophagus then relax, which means it is contracting. The contractions are repeated in a wave that moves downward to the stomach. Okay, this particular movement here. Eventually, the bolus, food bolus, is kind of conducted downwards. The cardiac sphincter lies between esophagus and stomach, allowing the conduction of bolus into the stomach. This should be gastroesophageal sphincter, not really the cardiac sphincter. The sphincter closes again, and medulla oblongata in the brain controls the peristaltic movement, the involuntary movement of the uh, like of the muscles in the stomach. And stomach is actually the widest organ of the alimentary canal. It is divided into two parts, two major parts. One is the body, and next comes the antrum. This is the body, this is the antrum here. Oral portion is the first two-third of the body, and caudal portion is the remaining portion of the body and antrum. So as the food enters in the oral portion of the stomach, it forms concentric circles. When food stretches the wall of stomach, vasovagal reflex travels from stomach to brain and back to the stomach, due to which the tone of muscular wall of stomach body reduces and the wall starts bulging out so that it can accommodate greater quantity of food. Now, in the completely relaxed stomach, 0.8 to 1.5 liters of food can be put inside. And after mixing the food with the stomach secretions, the resulting mixture is called a chyme. Okay, so it is this chyme is further passed down into the gut, and partially digested food, uh, basically partially digested proteins and fats, uh, take place, like partial digestion of proteins and fats takes place in this part, in the stomach. Okay, and it produces this intrinsic factor which is required for the absorption of vitamin B12 to be absorbed through intestinal wall. Next comes the small intestine. It comprises of three parts, namely duodenum. This is the small intestine here, okay, this part. So it is having the duodenum, the one which is immediately originating out of the stomach. Then comes the jejunum, and then comes this ileum here. So the length of the small intestine is correlated with the height of the person, but not with the weight. It is about 6.25 meters long, and it is the longest part of the alimentary tract. And chyme is conducted through the small intestine also by peristaltic movement, peristaltic contraction. Okay. So during movement of the chyme through the small intestine, complete digestion of proteins, carbohydrates, fats, and nucleic acids occur. Now here, this is the small intestinal beginning, the duodenal part. So here there is the ampulla of Vater present. You can see it here as well. This is the ampulla of Vater. So this is an important landmark in the biliary system. It's the place where the common bile duct here, coming from the gallbladder, and the pancreatic duct opens into the, like, merge into the small intestine. So ampulla is actually a reservoir that holds bile and pancreatic enzymes and release them into the intestine to help in food digestion. Next comes the large intestine, whose diameter is always larger than the small intestine, but the diameter can also vary from one region to another, and it is about 1.5 meters long. In terms of length, large intestine is actually smaller than the small intestine. And large intestine is again divided into three parts: the cecum, the colon, the rectum. And this is the colon. Colon is actually again having three parts: ascending one, transverse, and the descending colon. Then First is the cecum, then this colon with three variations, and then finally rectum where stool is stored. So cecum is a pouch-type structure. This one, the outgrowth of cecum is a vestigial body known as the appendix, or vermiform appendix. The cecum is more developed in herbivorous mammals than carnivores. The junction of ileum with cecum, this part here, the junction of ileum and ileum is part of the small intestine. So the junction of ileum with cecum is guarded by the ileocecal valve, and the function of this valve is to prevent the backflow of fecal elements from colon to the small intestine, ileum of the small intestine. The valve can resist back pressure of at least 50 to 60 centimeters of water. Then comes the proximal region of colon, concerned mainly with the absorption, and distal region with storage. The lower portion of the descending colon, this one here, this is the descending colon, is the sigmoid colon. Because of the shape that it has a sigmoidal shape here, and it opens into the rectum. It is 20 centimeters long and terminates into the 2 centimeter long anal tract. When a mass movement propels feces into the rectum, the desire for defecation occurs. The opening of anal tract is called anus. The anus has two sphincters, internal anal sphincter composed of smooth muscle fibers and external anal sphincters composed of striped muscle fibers, which are actually voluntary in nature. So the moderate quantities of vitamin B complex and vitamin K also found by bacteria are found here in the large intestine. Then there are several glands. The gastric glands, the entire surface of stomach lining contains mucus secreting cells. The stomach mucosa has two types of tubular glands, oxyntic cells, these ones here, oxyntic cells, and which are again gastric glands, and pyloric glands or pyloric cells, which the oxyntic cells secrete HCl, pepsinogen, intrinsic factor, and mucus, and pyloric gland secretes mainly mucus for protection from stomach acid. Also, pyloric glands, pyloric cells can secrete gastrin hormone. Now coming to the liver, it is the largest gland of the body and mainly secretes bile, normally between 0.6 to 1 liter per day. Bile serves two major functions. One is fat digestion and absorption. Along with the enzymes for fat digestion, bile acids in bile helps to emulsify the large fat particles of food into many smaller particles, the surface of which is attacked by lipase enzymes, lipid digesting enzymes, secreted in the pancreatic juice. So bile acids help in the absorption of the end product digested fat through the intestinal mucosal membrane. Also, excretion of waste products from the blood is done with the help of liver. An important waste product, bilirubin, which is the end product of hemoglobin digestion and excess of cholesterol, are also excreted out with the help of bile. A pear-shaped structure attached to the posterior surface of the liver stores 30 to 60 ml of bile, which is the gallbladder. Like this pear-shaped structure is the gallbladder, which is secreted by the liver. This one here, the gallbladder, pear-shaped structure. And then we have this pancreas over here, which is a soft lobulated large compound gland whose internal structure is similar to the salivary gland. It lies parallel to and posterior to the stomach. Pancreatic acini secrete digestive enzymes, whereas large amount of sodium bicarbonate solution is secreted by small ductules and larger ducts. The mixture of these enzymes and sodium bicarbonate passes through a long pancreatic duct, this one here, and this pancreatic duct eventually joins the hepatic duct, like with a common bile duct, before it empties into the duodenum through the papilla of Vater. Okay, so pancreatic enzyme and substrate. There are these enzymes trypsin, chymotrypsin, and carboxypeptidase, which acts on protein digestion, like protein molecules, and digests it into smaller peptides and amino acids. Then we have pancreatic amylase, which acts on carbohydrates. Then there are pancreatic lipase, cholesterol esterase, and phospholipase, which acts on fats. So here you see in the picture there are two sphincters. One is the sphincter of Boyden in this part and sphincter of Oddi in a little lower part. So sphincter of Boyden is the opening of the bile duct. This is the common bile duct into the pancreatic duct. This is the pancreatic duct which is guarded by the sphincter of Boyden, and it checks the backflow of bile. Then a little lower, sphincter of Oddi comes, which is the opening of hepatopancreatic duct into the duodenum, which is guarded by the sphincter of Oddi, and it checks the backflow of bile and pancreatic juice. Then there are intestinal glands which are formed by the modification of surface epithelium of small intestine, and two main intestinal glands are the Brunner's glands and this is the crypts of liver cone. So Brunner's glands are found only in the first few centimeters of duodenum. They secrete large amounts of alkaline mucus to protect the duodenal wall from highly acidic gastric juice, and it helps to neutralize the hydrochloric acid. Then there's crypts of liver cone, which are really like small pits located all over the entire surface of the small intestine, lying in between the intestinal villi. So these liver cones are covered by epithelium composed of two types of cells. One is goblet cells which secrete mucus, and then there are enterocytes which secrete water and electrolyte, also helps in reabsorption of water and electrolyte along with the end product of digestion over the surface of adjacent villi. And at the base are present the Paneth cells and Argentafin cells. Now, when we talk about the Paneth cells at the base of crypts of liver cone, these are secretory epithelial cells located at the ends of the intestinal crypts. The function of these cells is secreting antibacterial proteins into the crypt's cavity, thereby providing protection for the stem cells which line the crypt walls. Here, the stem cells are lining this particular wall, and here in the cavity, these Paneth cells secrete the antibacterial proteins. And then there are argentafin cells, which are also known as enterochromaffin cells. These are present throughout the digestive tract and mostly concentrated in the small intestine and appendix, mainly responsible for secreting serotonin, a hormone that stimulates smooth muscle contractions, which are essential for the peristaltic movements for helping the food move through the digestive tract. These cells are located randomly within the mucous membrane lining the intestine and in tube-like depressions known as liver cone glands. Presence of these cells is crucial for nutrient absorption, particularly vitamin B12, and for the overall function of the digestive system. So if we talk about complex food material like bread, which is rich in carbohydrates. So it is eventually having starch or polysaccharides which get broken down to finally give rise to the simplest sugar, the glucose. Then butter, the food that we intake, is a fat-rich food with long chain polymers of fatty acids and glycerol and glycerides. Eventually, the smaller and simpler molecules are formed upon digestion of butter or butter-like substances to give rise to fatty acids. Then we have cheese, which do have fats also, but mostly it's proteins, which are polymers of amino acids, and eventually peptides and polypeptides and peptides are broken down to give the simpler constituents, which are the amino acids. So in case of protein digestion in the stomach, there are two proenzymes which are not really functional, but they help in functional as in they don't directly act on the substrate, they help in activating another enzyme which eventually acts on the substrate. So there are two proenzymes, which one is pepsinogen, one is prorenin. Pepsinogen, upon in the presence of acidic pH, gives rise to pepsin. Pepsinogen is the inactive form, proenzyme, and then there's prorenin, again an acidic pH, and under the action of pepsin, it gives rise to renin. So, casein in milk is mostly in case of infants and children, Casein in milk is broken down to paracasein by the influence of renin, and paracasein plus calcium gives rise to calcium paracasinate. Now, in the small intestine, there are three proenzymes. One is the trypsinogen, chymotrypsinogen, and procarboxypeptidases. Trypsinogen is acted upon by enterokinase or to form trypsin. Chymotrypsinogen is acted upon by trypsin to give rise to chymotrypsin, and procarboxypeptidase is again acted upon by trypsin to give rise to functional carboxypeptidase. In the duodenal region, proteins plus peptones are acted upon by trypsin enzyme to give rise to peptides like smaller polypeptide chains. Okay. Casein is acted upon by chymotrypsin to give rise to paracasein as well, along with renin. Paracasein plus calcium again forms calcium paracasinate. Now, calcium paracasinate is again acted upon by chymotrypsin and trypsin to give rise to peptides and calcium. There's elastin and water acted upon by elastase enzyme to give rise to peptides. And large peptides plus water are acted upon by carboxypeptidase enzyme to give rise to smaller peptides. Finally, in the jejunum part of the small intestine, peptones and smaller peptides are finally acted upon by aminopeptidases to give rise to much smaller peptide chains and amino acids as well. So if we talk about carbohydrate digestion, starch is acted upon by ptyalin or salivary amylase inside the mouth to give rise to maltose, isomaltose, and limit dextrin. Uh, upon being acted upon by dextrinase, dextrin can give rise to glucose. Maltose in the presence of maltase by the action of maltase gives rise to glucose. Isomaltose again can give rise to glucose. And there's lactose. Upon lactase activity, it gives rise to glucose plus galactose. Sometimes too much intake of lactose can lead to lactose intolerance, leading to uh, the production of gas. Okay. So if we look at the starch or polysaccharide digestion right from the buccal cavity to the intestine, we have uh, we have starch, glycogen, or polysaccharides. In the buccal cavity, we already have maltose by the action of amylase or ptyalin. In stomach also we can have maltose. In small intestine, maltose, dextran, or isomaltose is finally converted to the simplest form, the glucose. Fat or long chain polymers in the stomach, fatty acids and glycerol. In the small intestine, it gives rise to diglycerides, monoglycerides, fatty acids, or glycerol. And polymer of amino acids, proteins, are broken down into peptones. In the small intestine, it's further broken down to peptides of different lengths to finally again broken down, to finally again breaking down and giving rise to amino acids. Then we have the nucleic acid, nucleotide sequence, DNA and RNA, which in the small intestine give rise to nucleotide individual building blocks, and eventually the nucleotides are broken down to give rise to pentose sugar, separately nitrogenous bases, etc. Then we have villi and microvilli, uh, which again helps in trapping of the food, passage of the food due to the microvilli present over here, even in the absorption of food as well. It increases the surface area of food absorption actually. So in case of absorption, what is happening? This is the lumen of the small intestine. Protein, different kinds of macromolecules like proteins, carbohydrates, fats are actually broken down to the simpler forms like protein into amino acids, carbohydrates into saccharides or monosaccharides, lipids into emulsified droplets, and free fatty acids, which are eventually taken in. It passes through the epithelial cells of the small intestine, taken in through these microvilli and stuff, and eventually get absorbed into the bloodstream. Now, in case of enterocytes, Again, there's movement of electrolytes as well, movement of the simplest sugar form as well through the enterocytes, and this is affected by the microvilli here, which increases the surface area for absorption. Now let us look at some of the questions here. Um, which process is characteristic of a saprotrophic nutrition in fungi? Intracellular digestion, extracellular digestion, phagocytosis, or autolysis. So in saprotrophs, what happens is they secrete some substances outside, the food is outside their bodies, and they secrete some substances on the food, digestive enzymes and stuff. Then take in that digested matter inside. Okay. So it's basically option B, extracellular digestion. So fungi release enzymes outside the body and then absorb the digested nutrients. So this is an assertion reasoning type of question. Assertion is Cuscuta is a parasite. It lacks chlorophyll and depends on host plants for food. So if both of these statements are true and one is correctly explaining the other, that's what we have to talk about. So Cuscuta is actually a parasite. It lacks chlorophyll and depends on host plants for food. So correct is like as in both statements are correct, and eventually this second statement nicely explains the first statement as well. Okay, because it lacks chlorophyll and it absorbs nutrients via hostoria, making it a true parasite. Which organism is an endoparasite? All these bedbug, mosquitoes, leeches, these usually are on the uh, on our skin. Whatever they do it on our skin. Okay, outside the bodies. But tapeworm exists inside our body. So correct option will be uh, endoparasite if is the tapeworm. Others are ectoparasites. Which statement about autotrophic nutrition is correct? Carbon source is CO2. Energy may come from sunlight or chemicals. It occurs only in plants. So carbon source is CO2. Energy may come from sunlight, photoautotrophs, or chemicals, chemoautotrophs. It occurs only in plants. No, it can occur in other organisms as well. Okay. So the correct option will be like the correct statements are the first and the second statements, but saying that it occurs only in plants isn't correct because there are algae, bacterial organisms as well, which carry out autotrophic mode of nutrition. So correct option will be option B, where 1 and 2 options, statements are correct. Chemoautotrophic bacteria like nitrosomonas obtain energy from sunlight? No. Organic food molecules? Fermentation? No. A correct answer will be oxidation of inorganic compounds. They oxidize substances like ammonia into nitrite. In insectivorous plants like Nepenthes, insects are mainly digested to obtain which particular element? It is nitrogen, where because these plants are growing in nitrogen deficient soils, so they actually are digesting the insects to obtain nitrogenous compounds from them. Which organism shows mixotrophic mode of nutrition, as in it can become autotrophs or it can become an autotroph or a heterotroph based on the surrounding conditions. Correct option will be Euglena. It performs photosynthesis under light conditions, when light is present, but becomes heterotrophic in darkness. Paramecium is heterotrophic. It also shows holozoic mode of nutrition. Rhizopus Cuscuta we looked at, it's parasite. Okay. So for mixotrophic mode of nutrition showing both autotrophic and heterotrophic, it is Euglena. Now, which organism shows holozoic mode of nutrition? We looked at one of the examples. I have the answer already written also. It's amoeba. So amoeba actually phagocytoses the food material, engulfs the complete food material and eventually digests it by secreting enzymes inside the food vacuole. Now, a patient has reduced secretion from the salivary glands but normal gastric and pancreatic secretions. During digestion of a carbohydrate-rich meal, which of the following patterns of starch digestion is most likely? Starch digestion begins in the stomach and continues in the small intestine. Starch digestion begins in the buccal cavity but stops in the stomach and resumes in the intestine. Starch digestion begins only in the intestine, and starch digestion begins in the buccal cavity and continues throughout the stomach and intestine. So the options straight away, the buccal cavity ones are anyways um, like because we have to discard them kind of because it has already reduced secretion from the salivary glands. Okay. Um, well, this one here. Okay, so the correct option will be option C because it has problematic secretion, I mean reduced secretion or no secretion in the salivary gland. However, normal gastric and pancreatic secretions are there. So starch digestion will begin only in the small intestine. So starch digestion normally begins in the buccal cavity due to the salivary amylase enzyme. Okay. If salivary secretion is reduced, then initial starch digestion won't occur in the mouth. Gastric pH, such a low pH actually inactivates amylase enzyme, so digestion won't even occur in the stomach. But starch digestion will therefore start only in the small intestine by the action of pancreatic amylase. In mammals, heterodont dentation. Heterodont dentation means the different types of teeth that are present. Okay. It allows for specialization of teeth for different functions, like we saw for incisors for biting and canines for tearing the food. So suppose a mutation reduces the development of premolars and molars while incisors and canines remain functional. So what type of digestive change is supposed to occur? Incisors and canines are there, but premolars and molars aren't really nicely developed. So what will happen? Increased salivary amylase activity to compensate for grinding, reduced efficiency of mechanical digestion leading to increased workload on gastric digestion, increased bile secretion to compensate for carbohydrate secretion, increased pepsin secretion to replace mastication. Mastication is the chewing of food and then forming the bolus along with the saliva. Okay. So, if only incisors and canines are present there, but the molars and premolars for, really chewing and grinding the food, those aren't functioning properly, then we will have efficiency, like reduced efficiency of mechanical digestion. Okay. Eventually, gastric digestion, the gastric enzymes are secreted, the gastric enzymes being secreted are the lone warriors in this digestive function. Okay. In case we don't have the teeth functioning properly. So the correct answer will be option B. Because premolars and molars are responsible for grinding and chewing the food, and loss of grinding ability reduces the mechanical breakdown of food, which results in larger food particles entering the stomach, hence increasing the reliance on gastric digestion. The researcher studies the digestion of a mixed meal containing starch, proteins, and triglyceride in the human alimentary canal. The following experimental observations are made. Hydrolysis of starch continues for a short time even after food enters the stomach. Efficient lipid digestion occurs only after emulsification in the small intestine. Proteins are initially converted into proteases and peptones, proteases and peptones before smaller peptides are formed. Which combination of enzymes correctly explains these observations? So basically, these are the three effects that we are seeing, and which all enzymes are doing this function that we have to match here in this question. So first one is starch hydrolysis continues for a short time even after food enters the stomach, which is acted upon by salivary amylase or ptyalin. So starch digestion begins in the buccal cavity by salivary amylase. Even after food enters the stomach, this enzyme continues to act briefly until the pH becomes strongly acidic, thereby affecting the activity of amylase enzyme. Next is if efficient lipid digestion occurs only after emulsification in the small intestine. Triglycerides are actually digested mainly in the small intestine by the pancreatic lipase enzyme, but efficient digestion actually requires proper emulsification of bile salts. Then comes proteins are initially actually hydrolyzed in the stomach by pepsin. producing peptones, which are later converted to smaller proteins by pancreatic enzymes. Correct option will be this one. Option A. So that's it for today's tutorial session. So we will again meet the next, sorry. Um, we'll again meet the next Sunday with the next week's tutorial session. Thank you for joining. I will end the meeting for everyone.